Hello everyone, my name is Murtra Enoch, and this is the continuation of revision BI 110. So, part 2. First question: suppose that the homologous chromosome that makes up a tetrad fail to separate during anaphase 1 of meiosis. What this phenomenon called, and what could be the outcome of an individual's 3 marks? So when they say tetrads, those are just the sister chromosomes. So we start answering. So the phenomenon, that is what we call nondisjunction. Nondisjunction, that is just the process by which sister chromatin fail to separate during cell division. So now this may results, or the effect of this same thing is that you find to say that it may lead to aneuploid. That is just the disorder where an individual has an extra or missing chromosome. Another thing is that it may cause trisomy and monosomy. Not only that, you find to say that it may also cause what we call Gardner-Neefter syndrome. That is just the genetic disorder where a male has an extra chromosome. So instead of a normal having X1, and then there is an extra X. So this condition is what we call Gardner-Neefter syndrome, and it may also cause what we call miscarriage. So these are the required solutions. So we proceed. So the next one, they are saying, name the processes that eukaryotic cell division and prokaryotic cell division, that is binary, have in common. So there is 3 marks, so I'm going to write B. So you find to say that in both eukaryote or prokaryote, you find to say that there is chromosome segregation, that is just the movement of the DNA molecules to the opposite pole. Another one is that in both eukaryote and prokaryote, there is cytokinesis, the splitting of the membrane of the cytoplasm. The splitting of cytoplasm is what we call cytokinesis. And then another one is that you find to say that in both there is DNA replication. So where the genes, they are copied the way they are. So these are the required answers, but you can also include if you want cell growth, because when both processes are occurring, they result in the growth of the cell. So we proceed to the next one. So they are saying, draw a cell in the following phases: mitotic phase, mitotic metaphase 1, and mitotic metaphase 2, and explain the differences between these 3 phases. So we proceed. So one thing that you should bear in mind is that under mitotic metaphase, we're going to draw the shape. So the centrioles, we do that. Also there, write the centrioles. These are chromosome. These are chromosomes aligning themselves in the metaphase plate. So also indicate so this is how the cell looks at the mitotic metaphase. So the single chromosomes are aligned themselves in the metaphase plate or equator. So, and then at the mitotic metaphase 1, you find to say that there now is different. You find to say that them is the homologous chromosome. Homologous chromosome are the ones that are aligned. So make sure that you draw the things that is visible. So homologous chromosome meaning two pairs of what? Chromosomes. So two pairs of chromosome, they align at the metaphase what? Place. Then you just indicate something like that. Something like that. So that is how the cell look at the mitotic metaphase 1. Now what about the mitotic metaphase 2? You find to say that there it has already been what? Separated. So you find to say that there is the single chromosomes that are aligned. So make sure you draw the thing that is very visible. So there is single chromosome. Single chromosomes. So it means that we have drawn the structure of the cell on how it looks at each stage. You can talk about the differences. So I'm going to put the differences the way they are. So when you look there, the alignment of chromosome, they what? So single. There is single alignment of chromosome. So chromosome, single chromosome, they are aligned. Now there, what you find that the one that are aligned are what? Homologous chromosomes. That is about the alignment. So there is single chromosomes are aligned. So when you write like this, you're talking about the alignment. What about the haploid? You find to say that there the number of haploid, that is diploids, and then on the other part you find to say that it is also what? Diploids. The last one you find to say that it's haploids. And then another thing that you can talk about, the aim of these the three phases that are being illustrated there. So you find to say that the aim of this is to produce identical what? Daughter cells. identical daughter what? Daughter cells. This one brings about genetical variation. Then this one it brings about formation of 4 haploid daughter cells. So writing like this, it means that you have obtained 6 marks. So we proceed to the next one. So they are saying chemotherapy drugs such as vincristine and coxine disrupt mitotic by binding to the tubulin or subunit of microtubule and interfering with the microtubule assemble and disassemble. Exactly what mitotic structure do these drugs target, and what effect would have on cell division? So the specific parts that are being targeted as spindle fibers, or you can say microtubules. So these are the structures that are targeted. You find to say that when these are affected, this microtubule has the tubulin protein, so those drugs they bind with the tubulin protein. That prevents these microtubule from producing spindle fibers. And then what will happen is that the effect is that the effect is that the cells become arrested in the metaphase stage, triggering apoptosis. So what will happen is that under metaphase stage, you find to say that when these are affected, the cell are arrested and they are prevented from undergoing cell division. Hence they are going to be forced to undergo apoptosis. That is just the process of those cells killing themselves. So please don't forget to subscribe, also share, so that you watch every time when I upload the new video. See you in the next session of our revision.